Good morning, everybody, and welcome to this bug meeting or the user meeting of ESRF 2022. Welcome to all of you who have already connected. We are 56 at present. So we shall start with a short introduction and reports from the beamlines. And then we will have a very short coffee break and a few more detailed instructions on the news and views of ESRF.
So yeah, I'm going to give a brief update on ID 23.2, and I'm just going to mention a couple of slides about auto processing. So I hope you forgive me for slightly going over my allotted time. I think, as many of you know, ID 23.2 is a microfocus beamline dedicated to MX, and some of the design goals were for stability, ease of use, so specifically no expertise in microfocus crystallography required, although it is helpful, of course, and a consistent user experience with the other ESRF MX beamlines. It's been in operation since 2006, and we've gone through various upgrades to essentially every part of the beamline downstream of the mono. And the most recent incarnation of it is shown at the lower right. Upper right is where we started. So now we have the same kind of sample changer as everywhere else. We did go through a significant upgrade in 2017 for optics, as well as diffractometer and other components. So I'm going to briefly mention some of those now. So this is the beamline layout. I'm only going to spend a couple of seconds on this because it's not so relevant to you. For vertical focus, we have two sets of transfocators after the mono, and then for horizontal focus, we have two sets of mirrors. And then the mirrors are in the experimental hutch as before, and that is of some practical significance because while we almost never open the optics hutch during the run, the EH obviously gets open and closed a lot. So those are very sensitive to temperatures. That's one of the things we ask users when you're here is to minimize the open time of the hutch. Okay, so one of the upgraded components of the beamline was, as I mentioned, transfocators allow us to rapidly change the vertical beam size, so in a matter of seconds. We have various beam sizes that are available in MX Cube. Horizontal focus is a bit slower to change, but we can switch between the two mirror systems. So if you have very small samples, very small meaning less than 5 microns, you can tell us and we can optimize the beam down to around 1.5 if necessary. The Diffractometer is a new instrument, relatively new instrument, allows us to do very fast mesh scans, has excellent on-axis viewer, a split beam stop, which allows us to move it downstream of the sample or upstream of the sample as required. Various apertures, which in our case are used only for cleaning the tails. The focus is largely achieved just through the properties of the mirrors. But they do cut down on the tails, so they're useful. The beam visualization is quite convenient, and that can be important, as I said, if the hutch has been opened and closed, you should be paying attention to where the beam is, and that's easy to do. We have a mini kappa. So previously, we were not able to use the kappa because of the sphere of confusion, but with this vertical orientation, it actually makes no difference whether it's on or off to the sphere of confusion. So we leave it on. Which is a nice feature if you have a large unit cell, for example. We have a plate gripper, and hopefully we'll be making some more progress on a version of MX Cube that takes full advantage of that this spring and summer. And it's been used as a test bed for various serial devices in the past year, which I'm not going to talk about today. But if you have devices that you'd like to test, please contact myself or Shibam. We have there's the scanning done in real time. We have the Flex HCD, and I'd just like to remind people that it's unipux only and spine samples only. So if you use the crystal cap, you will cause a collision in the doer, which can be a big problem in full remote mode. So just please make sure that everyone in your labs is not using solid crystal caps. Okay, so I'd just like to add a couple slides on some interesting recent structure solutions. One was a 200 amino acid protein, 8 protomers in the AU, C221 with 2.9 angstrom diffraction. And we tried both Archimedes light and AlphaFold to try to phase this thing, and neither of those worked. And actually, one interesting thing that I found in a very recent paper on using AlphaFold for molecular replacement is that coiled coils, first of all, are not, well, they can be difficult for AlphaFold to correctly predict, and also coiled coils, I guess, are inherently difficult with molecular placement. And that proved to be true for this protein, which we knew to be a coiled coil, so we ended up solving it actually on 23.2 with selenium met substituted protein. I should mention that it actually happens, although it's not a tunable beamline, there's enough anomalous signal at 14 keV to phase proteins fairly routinely. There's still 3 electrons of F double prime. The trick generally is to do a very high multiplicity data set, which is what we did here, helical data set, which ended up with about 8-fold multiplicity and very nice stats. Although there were quite a lot of selenium mets, only 12 sites were found, and the peak heights were actually not fantastic. There were about 6 to 11 in terms of the peaks that were found, and you can see it is indeed a set of coiled coils. And you know, I think this is emblematic of cases that will be difficult for AlphaFold 2 to solve, coiled coils, many in asymmetric unit, not super high resolution diffraction. So 23.2 is not was not conceived for phasing, but we also had kind of a fun test experiment last spring. We had the typical test crystal cubic insulin, and remember we're at 14.2 keV, so the F double prime is 0.18. We used the small beam setting on a fairly large crystal, but a very massively high multiplicity helical data set to 1.2 angstroms, so 90-fold multiplicity, very high I/O guys, and we're actually just able to solve it by sulfur SAT, finding all 6 sites with actually quite impressive anomalous peaks. And this is not to say this is the beamline that you should be doing this on, but it's mostly meant to highlight that we get very high quality data, and you can really boost things by doing helical. So I do recommend people do a helical as much as possible. You don't want to go too large typically, because crystals do vary within the crystal, so you should understand how sort of isomorphous things are within your crystal before you go to a very large line size. And this was these data have been uploaded to Zenodo, and it's been submitted to the PDB. I'm trying to submit the unmerged data, but it's been breaking things because of the high multiplicity, but it will be there. Okay, so in terms of improvements, we'd like to enable the ROI mode. We'd like to use the mesh scanning data to actually produce data sets. Shibam has already written code to do this. It just has to be rolled out and tested a little bit more. And right now we have automatic cluster analysis and genetic algorithm analysis of mesh and collect data, but it needs to be improved in terms of reliability, but it is there. Okay, so a few words about auto processing. We recently got more compute nodes added, so that's allowed us to increase the number of nodes devoted to each process, speeding things up. As I mentioned, we have automated processing of mesh and collect. We also have a much more sophisticated addition of ligand files. So you can specify multiple ligands in single files, complexes with different components. All of those are supported now in via XE. We also added AlphaFold. So if you provide a uniprot ID, first we check for that uniprot ID in the PDB and use that for MR. And we also check the EBI AlphaFold database. And if there's a model there, we use the entire model to, well, we trim down the low probability regions and use that for molecular replacement. And we also use Phoenix process predicted model to break it into domains, and then each one of those domains are then submitted to MR. Of course, eventually we'd like to be able to, at the upload time or at specification time, say which parts of your AlphaFold model you want to use. But of course, you can always just upload that model directly if you want at the moment. The way you specify it is quite easy. You go to proteins and crystals, select my proteins, go to your previously described crystal forms, add a structure, and then the rest should be automatic. And I should mention also that this can be done at the very last minute because it's using the EBI database. It's not actually calculating a new AlphaFold model. You could even do it during game time. Okay, so if it works, then you get the typical green bar that some of you might have seen for various kinds of phasings. This is a real-world example of reasonable data, I guess. What you'll see is if you drill down a little bit, you'll see MR from AlphaFold, and then you can click and see density like that. So that's pretty much all I have. If we want to move on to the next person. I don't know if we're going to do questions now or later.
I think we will move all the questions later for sake of time. Thank you very much, Dr. Nanau. And now we will go back to the first speaker, Dr. Sasha Popov.
Oh,
Can
yeah,
you share your screen?
Yes, yeah. I'm sorry,
No problem.
Had problem with my computer. Yeah, so it's can't restart. It's true. Happened. Just a moment. Are you saw my presentation or you? It's
we see the notes.
Okay. So I need to. What I should do? Yeah, like
yes,
this, yeah?
That's fine. That's fine.
It's okay.
Perfect.
So,
thank you very much.
So briefly about the what happened with 23.1 nearby. So during last year, it was completely new experimental setup. So first of all, we changed the diffractometer. So now we have new MX2 as micro diffractometer, which was mounted in April last year, which is nice diffractometer. We got some problem with this in the beginning, yeah, but now it works more or less okay. So we can provide quite fast rotation, scan, and so on. And of course, what's very important, we change also our detector. So before it was Pilatus 6M, and now we have excellent new Eiger 6M detector with a cadmium Taylor the windows, which provide us ability to collect data with using high energy, high X-ray energy. So, so we still have quite big. It's compared maybe with the other beamlines. It's not so big intensity, but we can change. We can select the X-ray energy from 6 kV up to 20 kV. And if we're going for the high energy, we still have quite good intensity. And what also important to mention, the divergency of the beam is improved when we go for the higher energy. And now, when we talk about more or less optimal energy for standard data collection, and we now recommended to go for 16-14 kV. Before it was about 12, about selenium age, but now we can recommend it to collect data using more higher energy. And this is one of the example how well our data can be. This is data collection from thermalizing. As you know, it consists of zinc. And using 20 kV and very, very big anomalous signal from the zinc, it was still possible to automatically salt SAT data, as you can see from this ShellX picture, is a clear solution and yeah, nice map which was constructed after ShellX. It's not ARP-warp, no refinement, simply map after ShellX. So this is main news about Beamline. So what I would like to say, we also try to go and also implement MXpress on the Beamline. So I hope soon we can propose users who would like collect data completely automatically. Its options also will be provide for 23.1. So this is one of the example which was recently, so a few days ago, we tried to see how it works on the 23.1. So still there are some small problem, but generally it's possible to use. Yeah, this is one of the example. And what I would like to say about the development, you know, when we're talking about this complete automatic data collection, one of the central problem, it's recognition crystal position, and usually we're doing it using so-called X-ray centering, and this we create special program which called Azor, and the program can recognize the diffraction where is this there is the point which provides the diffraction, and then also the Azor M program can recognize the best position from the diffraction, and also recognize is it crystal, is it one crystal or is it several crystals, there is some superposition of crystal position, and so on. And so we now try to develop a new program which called Azor M2. This is kind of continued development of this recognition, X-ray crystal recognition approach. That's why it's important. You see, we from time to time have this situation because usually we collect data, do one mesh scan, and then rotate crystal for 90 degrees, and using this selected best point, making the vertical linear scan, and select the third coordinate of our crystal. But quite often we have situation which is shown in this picture. So we have, for example, superposition of two crystals, or one crystal simply blocked for two parts, and we have problem how which of these two peaks belongs to the crystal which we recognize using this two-dimensional scan. So, and what we can do here, we also can repeat this two-dimensional scan also after rotation from 90 degrees, and using this new program recognize which position belong to the first selected crystal, or recognize two crystals, because from time to time we have this complicated situation when a crystal, like in this example, split it for several parts. So, and potentially we can organize our data collection to do several data collection using one part, and then a second part, maybe third part, depends from the situation. Because if we not properly select the second position, we can potentially have situation which is shown on this picture. So we collect only partial data, but most of the data during our rotation will be for nothing because it will be no crystal in this position. So this program already in the test, and there is the beta version and shown here. So this is one of the example. So we can do 2 scan, and then program automatically proposed the motor position, which will allow us to put the best part of the crystal or crystals and collect data. So, and what's important also, if we talking about mesh and collect data, when we have many small crystal inside the loop, and we can quite limited to we can't rotate our loop for 90 degrees because it's a physical limitation. And the problem to determine the third coordinate from our crystal, and this is new program, also will allow to solve this problem. We simply should collect 2 mesh scan separated for, let's say, 40-50 degree, and program automatically should recognize which crystal belong from which position and provide the net of coordinate, and then we can collect data using the standard mesh and collect data, but not limited our rotation range, so we can use bigger rotation range and provide a bigger completeness from each small crystals. So this is what we are trying to implement now. Hope it will be soon available for users. So that's all what I would like to say. Thank you for your attention.
Thank you very much, Sasha. Also for you, we will take the question at the end. And now it's the time to switch to Dr. Igor Metnikov.
Hello, everyone. This is the yearly update for Massive 3. This is just to recall what the Beamline Layout is. We use, this is the optical setup. We use lenses for vertical focusing and KB mirror for horizontal focusing, taking a fixed energy out of silicon single crystal monochromator. So finally, we update, we obtain around 12.8 keV energy and 15 micron, roughly 15 micron beam in diameter at sample position. Here you can see the image of the beam profile at sample position and at horizontal focus. So as you can see, the beam is slightly overfocused on at sample position, and the measurements done with the optics group, they confirm that the size is roughly 15. So here at focus, it goes to even 3 microns. But we see that it's very convenient to use the round-shaped beam of 15 microns. The experimental setup looks like this. So we have standard equipment of our structural biology group, flex sample changer, MD2, diffractometer, and Eiger for detector. What's particular about IG4 is that it's quite fast. It's capable of doing fast data collections, and the minimum exposure time is 1.3 millisecond. It can be extended, of course, using the region of interest mode, but this will sacrifice the resolution quite a lot. So the real limit is 1.3 millisecond normally. And so this limit is quite, let's say, is not sufficient to record a lot of interesting processes in the biological world. However, even some states of the photocycle of photoactive proteins can be recorded. And what's also quite interesting is that what can be quite promising is the time-resolved studies for incrystallar reaction, incrystallar catalysis. Because the rates of the enzymatic reactions vary quite a lot, and many of the reactions can be indeed recorded using this, even at this time scale of 1 millisecond, roughly. A good example of this used so-called caged compounds or caged substrates, as presented here. The principle of this caged substrate is that it can be released using ultraviolet illumination. And then the reaction starts and we can record it. Here I put a very good example of such study. So binding of the substrate could be recorded, and time frames of the reaction as well. Another development available on the Beamline is online microspectrometer. So it can be used simultaneously with X-ray data collections. The optical spectra of crystals can be recorded. The principle is simple. So we just focus excitation light on a crystal and then record the what's been transmitted. In real life, it's a bit more complicated, and but still give nice results. This is done with a great help from the ICAS lab. And finally, we sacrifice a lot of effort to the development of crystal localization techniques on our beamlines. So one of the latest development is doing a double mesh scan instead of standard X-ray centering, as you may know from our beamlines. The advantage is, so the principle is like the X-ray centering. You just basically do a second mesh scan instead of line scan, 90 degrees apart, or it can be lower difference angle. Actually 20 degrees is more than enough. So you do two mesh scans, and the feature here is that we can now detect which crystal is where on these two maps, and then calculate more precisely each crystal coordinate. This will allow us to avoid such things where we have a crystal. Quite clear example here. Was centered, so the crystal was centered on a mesh scan map, and a line scan was performed then. Maybe it was quite clear from the start that it's right, but it was quite wrong. So data collection went not very well, and it was not properly centered. And such technique that we can detect whether it's the same crystal on the second map or not will allow us to avoid such situations and give more automation to our beamlines, and of course partially to Massive 3. And with all that, thanks everyone for your attention.
Thank you to you for all this very great advancement. Now we move on to Andrew Mccarthy. Andrew, are you there? I can see you.
I'm here.
I can see you.
So I'll just give a short update on ID30B on behalf of Christoph and myself, who are the beamline responsible for the beamline. And you can find more information, of course, on the website and also in the paper from a few years ago. So just a brief update on the current situation. So basically we have one U35 undulator. It's a tunable beamline from 6 to 20 keV. The current beam size is around 30 microns, and there's a lot of flux, so you need to be a little bit careful. So there's a flux around 10 to the 13, which is equivalent to something like 15 megagrays per second. So you need to be careful with the attenuation you use. So normally we would recommend anything between 1% and 5%. If you go above that, there's a likelihood that you will start to damage the crystal. For the experimental setup, we have the MD2S, which incorporates both the mini kappa head for crystal realignment and also the plate manipulator head, which can be swapped in something like 20 minutes. So if you're interested, just let us know. We have a flex sample changer with spine and unipuck. We have a Pilatus 36M with a 100 micron silicon sensor, which means you can collect high resolution data by changing the energy to 20 keV. And then software, we have the latest version of this, MX3, the Beamline expert system, which are all the automated workflows, how we operate them, and extended iSpy-B, and you'll hear about these later this afternoon. So the biggest change this year that we spent quite a lot of time on was actually stabilizing the monochromator, particularly after energy changes. So now we have quite a stable beam. So basically it stabilizes as soon as you change the energy. And this has resulted that the energy change is much easier now. So basically you can change the energy, which takes about 2 or 3 minutes. Once that's done, you can start your data collection or your X-ray fluorescence or X-ray energy scans for phasing. We also have a silicon stripe for low energies, and I'll talk just to show you a recent example of how we use that. And as I said, for higher resolution, you can make use of the 100 micron silicon sensor to collect data at something like 0.7 angstrom at 20 keV. So what's now available is that we have as I said, it's tunable, but we also have a variable beam size. But we do this by using the apertures. So we have a 10, 20, and 30 micron aperture. And basically we reduce the flux a little bit, but there's still quite a lot of flux at 10 micrometers. So you can really make use of that, particularly for X-ray centering. The experimental setup, as I said, we've changed a bit. So we have much more unipuck positions. So we took out 2 of the spine pucks, but we still allow these file spine pucks to be accommodated on the beamline. We have the MK3, and we have some crystal realignments using the workflows. We have the in situ crystallization plates, as I said, and we also have a dehydration device or for room temperature experiments as well, which are also quite useful for either improving the diffraction or just collecting data at room temperature. And as I said, we have most of the automation workflows available, particularly the MXpress workflows, and you'll see a talk about that from Didier and Matthew this afternoon. And we've been working quite a lot. There's a few industrial users who use these in automated mode, so it's been working quite well recently if you want to try it out. Just a little bit about the plate manipulator. So we finally configured it with the help of Jean-Baptiste and Matthias, or Marcus and Antonia from the Bliss unit. And Nicholas has been developing this at the moment. And basically allows you to take different types of plates. So we allow in situ, Crystal Direct and the Crystal Quick X, and you can navigate through the plate. You can collect data using a small oscillation range because it's not perfect centering, so you can only collect maybe 5 or 10 degrees at most. And we're working on automation methods to make this easier to use. And if you want to see more about that, you can see Nicholas has a poster presentation in SB5, which is in poster session 2, and he can, if you want to find out more about that, you can talk to him or see the poster at some stage. This also allows us to use the plate manipulator for microfluidic chip data collection, where we had a nice collaboration with Gavi in Granada, where they developed these counter diffusion in microfluidics plates, and Estelle is leading the developments at the ESRF at this moment. So if you're interested in trying some microfluidic chips, you can talk to Estelle. And it allows lots of different opportunities, such as ligand screening, room temperature data collection, etc. So as I said, we also have the dehydration device, and we have the Rex nozzle changer. So basically you can either have it in the cryo position or in the dehydration or humidity controlled system. So just let us know if you want that. We don't have it installed all the time at the moment, but we have one available if you need to try it out, or you're interested in trying it out, just let us know. And then just a quick recent highlight. So this was a little bit before the AlphaFold craze, but actually what we were able to do is to solve a structure from Raman Ravelli's group, which is unpublished. And basically we used molecular replacement to solve one of the proteins, but then it was very difficult to solve the other one, particularly because the data was low resolution, around 2.9 angstrom, and also it was highly anisotropic. But by using a combination of sulfur SAD, we were able to use these as markers to be able to align the sequence or to build the sequence into the protein structure. And we did that using 7 keV data and a sulfur SAD data collection using the mini-kappa as well. And also we used a mercury derivative in order to identify cysteine to help with the model building of the new protein that was built. And then just a little bit about what's coming soon. So we hope to really make use of the new X-ray source, the EBS. And what we're going to do actually at the end of this week is explore the possibility of using these 2D lenses. So for the horizontal focusing at the moment, we use 1D beryllium lenses, and we use a mirror for the horizontal focusing. And what we're going to do is to try and combine the two to have a 2D beryllium lens to focus both in the horizontal and vertical direction. So we're looking forward to trying these tests later this week. And then in the experimental setup, we want to further improve the in situ data collection. As I told you, we want to try and automate that a bit more so that people can actually set up an automated data collection. And as I said, you can see Nicholas about that. We want to try and re-implement the global phasing crystal realignment. So we have some in-house developments that you can run through the workflows, but we would like to also make use of the global phasing developments as well. And again, you'll hear more about that from Serena's talk. And lastly, we want to implement the Raman spectroscopy in collaboration with the ICOS team. So hopefully towards the middle or the end of this year, we can start to do those developments as well. And basically, it's a great thank you for everyone involved. and it's a good team to work with. And I guess we'll answer questions a bit later.
Yes, thank you very much, Andrew. Even here, a lot of new things are coming on all the beamlines. And now it's time for some SACs with Mark Tully.
Right, so good morning. I'm Mark Tully, Beamline Scientist on Beam 29. Our dedicated BioSACs beamlines, which I jointly operate with Petra. So for any new users here, I'll briefly just explain why we do SACs, the beamline, our software, and then just outline what is new and improved, and some information for users at the end. So why do we use SACs at all when we have our lovely high resolution techniques such as crystallography and cryo-EM? Well, SACs is a high throughput technique which can directly measure in the solution state. So we can then generate our conformational states and assemblies of material applied at a low resolution. So SACs is essentially a very complementary technique to all structural biology, whether that's MX, cryo, molecular modeling. And we can now actually quickly and easily validate our AlphaFold models too. Some examples of this are subunit organization, low resolution shapes modeling. And we can also use it to look at ligand binding as well. Our experimental setup has two sample environments, our robot and our side exclusion chromatography HPLC setup, where we can actually directly purify our sample before measurement. This is my preferred way of measuring. We also have a Pilatus P32M now, which hopefully in the next year will be going into vacuum as well. So a standard BioSACs experiment will take about 50 microliters of sample, between half a mg per mil and 5 mg per mil. Store now 96. We've got 96 watt plate and PCR tubes, and it's all thermoregulated between 4 and 40 degrees. Our measurements are all in vacuum. We've moved to a 1 millimeter capillary, so this speeds up our flow of liquids, but actually decreases our radiation damage. So even with the EBS, we're actually seeing no increase in radiation damage than before the EBS. We will also be hoping to put a UV across our capillary as well in the near future. We now have a second exposure unit, and so this has got a nice XYZ stage, and it's got a front light mirror. It's got a hole through the mirror, so the X-ray can go through the mirror. And this is actually what we're going to be using, and what we're developing now is our microfluidic system, so in vacuum microfluidics for SACs. Although I won't go much into that because this will be in Anton Popov's talk later today. So here's some of our data post the upgrade. So it's actually, we can see we've got very nice clean data. This was a protein which we had like three crystal structures of the domains, but we're actually then looking at the domains together. And you can see the together actually look very nice and actually fit into our models very nicely. So with most BioSACs beamlines now all having the automated sample handling robots and the HPLCs, what actually sets us apart from all the other beamlines now is actually our user software. So as many of you actually have already seen now, we've completely rewritten our collection software based Xcube. So it's been designed to be completely user-friendly and intuitive to new users. You can simply just click on your sample environment on the side, and then we have a nice queuing system, so you can actually just queue up sets of samples, whether that's in your batch or in your sec stacks. We've made it nice and simple. It's literally just click where the sample is, give it a name, add to the sample table, and run. And it's as simple as that. And obviously, there are little tabs, so you can actually expand that for if you've got more complicated experiments. So it's all in there, but it's just hidden for users. Recently, we've now just completely integrated our SECSACs into here as well. So this is now working. We simply just click this button here, and then our VSX will actually control the SECSAC as well. Again, just show where your vial is, and just give it a name, and you just put the number of frames you want to collect, and just run it and just add it to the queue. Again, very nice and simple, and will just help with running samples, multiple samples during the day. We've also changed our data processing software. So we've moved, our pipeline is now run by FreeSAS. So we've taken out AtSAS, and so we now run FreeSAS. It gives us all the same data as before, and is then integrated again into iSpy-B. So as this is a bag meeting, I thought I'd just quickly go over some user information. So we're now actually open to in-person visits, and actually we prefer it if you did come. Really mail-in mode, you can still use that, but we can only really measure 8 to 10 hours worth of samples. So obviously if you have multiple users coming from your bags, sometimes you cannot accommodate all of them. So please come back to us, and we'll actually help you, teach you how to use the Beamline, and help you reprocess data and help you analyze your data if necessary. Our most asked question at the moment is, how do you download your data? So we've now improved this. So firstly, we have iSpy-B. So we have these little down arrows where you can actually download your data. So this download all your DAT files and your averaged files in the which is being processed. And for the SECSACs, you just put in your how many frames you want, and then go to download range. And again, this will download all your .dat files, which then you can use to reprocess your data. If you want your raw data, then we've got the Data ESRF portal. This now has SilkView embedded, so you can actually look at your data here, and then you can actually download your H5 files, which contains all your raw data and your raw images. The larger files, or we can then use Globus, which again, handily, it shows you how to download and use this in Confluence. This actually has the added benefit that when you're in here, you can actually see all the folders and download all your data. So all your reprocessed DAT files and your UV data and everything, they can be all downloaded from Globus. So that's just a quick summary of what we've been up to this year, and just to thank everyone that's worked on our Beamline. Thank you.
Thank you very much, Mark. And a great thing that we can come back soon to the Beamline. And now the floor is to Isai, Dr. Kandia.
Hello, everyone. So I'm talking about the cryo-EM beamline in ESRF. It's CM01. And just a few latest news from this beamline and how we are operating so far. So what we have in CM01 is a Titan Cryos G3, which has an energy filter attached to it. And recently in June 2021, we upgraded the camera to a K3 direct electron detector camera. And we also have a Volta faceplate for very small molecules. So what we do in CM01 is only single particle experiments, which have proven to go to really atomic resolution. The access is either via bag or rolling access at any time. We also started giving rapid access for COVID proposals. And typically it's a 2 days experiment. And the beam time is granted by BTAP. Now for COVID proposals, this will not be reviewed by BTAP for scientific merit. So please contact the user office if you have a COVID project and you need cryos beam time. And again, for all the experiments except COVID, it's only pre-characterized samples where the sample preparation conditions have been established for very good eyes and particle distribution. And a preliminary data should have been collected at their home microscopes to provide proof that the 2D class averages can reach sub 10 angstrom resolution, which means that the cryos can provide high resolution data. Since 2017 November, we are operating, and it's about 4 years. So we have welcomed users from almost 16 different countries, and the feedback is not so bad. We have very good publications in high impact journals, and this is just a gallery of few examples covering macromolecules from viruses or viral proteins to membrane proteins. So I just picked one example, which is the latest publication from CM01, just to discuss this here, to show that how cryo-EM can help. So this is about the catecholamine synthesis. They are nothing but a group of amines, so which are very important neurotransmitters such as dopamine, adrenaline, or noradrenaline. And apparently they all are synthesized in the same pathway, with dopamine as the precursor for adrenaline or noradrenaline. Now, the first enzyme in this pathway is tyrosine hydroxylase, which converts the tyrosine to dopa. And once there is too much of neural transmitters, namely dopamine or adrenaline in the bloodstream, the dopamine is inhibiting the tyrosine hydrolase as a feedback mechanism, feedback inhibition. And then to restart the process again, there is a phosphorylation of a serine 40, which restart the tyrosine hydroxylase. Now, by crystallization, people have understood part of the process by crystallizing some of the domains. It has 3 domains, the enzyme, some of the domains with or without the substrates, inhibitors, etc. But still the whole structure was not, the full length structure was not available. So thanks to cryo-EM, now this group of scientists, they could solve the structures of 4 different states of the protein, namely the apo-TH, the TH bound to dopamine, and the TH where the serine is phosphorylated, and also various mutants to show that all these processes are controlled by just an N-terminal helix, as shown here. So this N-terminal helix, apparently it's folding back onto the active site where when dopamine binds, where it inhibits the enzyme. And then the serine 40 is located in this N-terminal, and when it gets phosphorylated, it opens back. So this is a very good example of how cryo-EM can help to understand the full mechanism of a neural transmitter synthesis. So now coming to the update news from CM01. Since June, we also did a microscope software upgrade, which allows us to do what is called a multi-grid options. It's somewhat similar to a crystallographic queuing system. So where you can queue several grids, you make a queue of data collection of several grids, and then everything goes online, the loading, unloading of the grids and the data collection of the grid in the stage. So this is especially very useful for drug screening purposes or epitope mapping, as shown in this figure. So for example, here 12 structures have been solved within very short time. As you can see here, each data set was only 4 hours. This is on the spike protein, which is a symmetric molecule as well. So this is a very good example, but needs to be optimized for real examples. But as I said here, it needs a well-established sample preparation like the spike protein. and also data processing methods. So, but recently we have tried here for our in-house research that we can also try this for grids with less acquisition area. Some proteins will never get to very nice grids. This could be proved. And if such cases, we can try for multiple grids on several grids of the same sample. But then we have to combine the data from several grids. So this is not same as the epitope mapping. So in this case, data processing must have been established where they can show that they can combine the data from several grids and go to high resolution. And when we opt for a multi-grid, this is a fully automated data collection where there is no manual cleaning of the selected area is possible. So which means the grid should be, or the grid squares should be similar and clean. So we apply a common ice filter on the same, on all the grid squares of the grid. So this is better if the grid squares are similar. So also the K3 upgrade led to a higher throughput. So recently we also, we were proposing to users since June at a maximum speed of 400 or 450 images per hour. But recently we characterized another mode in K3, which helps us to go up to 900 images per hour. This is, as I noted here, it's an ideal situation, which is an apoferritin, a very high symmetry molecule, nice grids you can obtain from this, 1 second exposure, not too many frames. And it's a bigger hole size where you can put multiple images, like 4 images per hole. But we also tried for some users already where we can put only 1 image per hole, and this goes to 500 images per hour. So we easily see with this mode, we can go anything from 1.5 times to 2 times speed increase. So which means we can collect up to 10,000 images from overnight data collection. So this together with the multi-grid option, it opens up several possibilities. As I mentioned before, for drug screening or inhibitors or epitope mapping, etc., we can do multiple grids of the same sample. We can also do multiple samples of the same user, especially for bag people or rolling access people. Also, we can also, like in MX, we can combine users of the same bag. At least 2 users we can combine to put both the samples from both users. There are few restrictions here, namely the total dose should be similar, so that the gain reference is done only 1 time. So it should be applicable to all the samples that's been scheduled for that 48 hours. So we will soon announce this to all the bag members and all the cryo-EM users. And perhaps we might contact you for some friendly bags. So we started pure mail-in experiments since, I think, since COVID confinement. So we have collected few feedbacks. For practical reasons, I think pre-screening of grids is really important now, because the local contacts can quickly organize and start the experiment as fast as possible. So the atlas is preferable. It's not obligatory, but if you have atlas, if you could do an atlas, please do so. And also, if you can lead us to where to collect data, you can send us a PowerPoint of the atlas and mark the areas that you want to collect. We could go ahead with that. It's much easier. And of course, the 2D classification is mandatory, and we will contact, the local contacts will contact the users to get the above details, and please make some earlier response so that we can quickly organize the experiment. And also earlier shipment is mandatory, because lately we had so much of cancellation of cryos time due to the shipment not reaching us. So one last slide to show that we will have a new cryo-EM beamline, CM02, which will function as a CRG beamline. It's from the French National Initiative, and it will have the latest cryo-EM microscope, perhaps with the energy filter and definitely with the direct electron detector. And what is the effect for CM01 users is that we will have 30% beam time from CM02. So this will help us to dissipate the users from CM01 to CM02, as the wait time in CM01 is quite high still. And it also permits us to perform cryo-electron tomography experiments. So I think that's all from me. So I thank everyone who helped, and thank you.
Thank you very much. Great things ahead, also for the cryo-EM side.
During this talk, I will give an overview of the high pressure freezing laboratory for macromolecular crystallography. Then I will give a few examples of use, and at the end, I will give some example of some general information. Sorry. So the high pressure freezing laboratory is proposed to users in complement to X-ray diffraction beamline, and it allows for the preparation of protein crystal under pure pressurized gases. If you have a look inside the PDB, you can see that there is already about 1,500 structure deposited in which there is a light gases. This shows the importance of studying light gases in structural biology. And the high pressure freezing laboratory aims at preparing this gas derivative. This slide describes the possible use of high pressure. High pressure can be used to map cavities and tunnels within protein, or it can also be used to when you pressurize gases like oxygen to study the reactivity of enzymes that are oxygen sensitive. High pressure can also be used to explore the structural conformation or fluctuation of protein. From the methodology point of view, high pressure can be used to freeze protein crystal without cryoprotectant, or it can be used to produce noble gas derivative for structure determination. The process of high pressure freezing is based on the same concept for all the gases we use. It is based on the thermodynamic property of the different gases. So first, the crystal is pressurized inside a tube at room temperature in order to produce the derivative. And after 10 minutes of soaking, about 10 minutes, the crystal is suddenly flash frozen inside the tube while it is still under pressure. And thereafter, the crystal is depressurized and handled in liquid nitrogen and stored in liquid nitrogen. It's important to mention here that we are using specific HP pin that are spine standard and also EMBL pucks. And that we are able to freeze a maximum of 4 pucks per day. The high pressure freezing laboratory is located in sector 30, just in front of Massif 3. Inside, users can have access to a sample preparation bench, a fume hood, the different pressure cells that are controlled by computer. And we are currently installing a system of glove bags in order to handle oxygen-sensitive crystals. This slide describes the different pressure cells which are available. We have a pressure cell to freeze crystal under 2000 bar of helium without cryoprotectant. This system can also produce argon derivative we have a cell to produce krypton derivative under 150 bar of pressure, and we have an enlarged version of this cell, which can produce krypton derivative under 500 bar. We have a cell for pure oxygen, and we are currently installing a pressure cell for carbon dioxide. I will say a little more about this at the end of the talk. Concerning examples, the first example of use concern here an alloalkane dehalogenase, which is an enzyme that is able to catalyze the cleavage of carbon halogen bonds. This enzyme can be used in bioremediation processes. So the users have designed here an improved mutant of their enzyme using a program called FireProt, and this mutant has display an outstanding thermostability. So the structure of the mutant was compared to the structure of the native in order to understand the structural basis of this hyperstabilization. In the native, the buried active site is connected with the solvent via two tunnels, which are essential for the catalytic activity. And in the mutant, surprisingly, the tunnels are blocked or occluded by the mutated residue. So in principle, the mutant should be inactive. Krypton derivative of crystal of these mutants were produced at the high pressure laboratory in order to probe the accessibility of the active site of the mutant. And you can see here in the anomalous map that there is a series of 12 krypton within the channel, which means that the active site is effectively accessible. So the outstanding thermostable properties of this mutant were explained by the fact that at higher temperatures, the dynamics of the protein increase, and then the tunnel are reopened, and then at high temperatures, the mutant is fully active. The second example concerns another enzyme, urate oxidase, which catalyzes the oxidation of uric acid, and this enzyme is used in drug treatment against hyperuricemia. The enzyme was crystallized with an inhibitor, and the crystal of urate oxidase were studied at different pressure from atmospheric value to 2000 bar under argon. At atmospheric pressure, the molecular assembly display an homotetramer, and you can see that the pressure induces or reveals the first step of the dissociation of this homotetramer into homodimer. The pressure also induces some modification in the active site at atmospheric pressure. A single inhibitor is observed in the active site. At medium pressure, an argon atom rapidly saturates an hydrophobic cavity next to the active site, which serves as a transient reservoir for oxygen. And at high pressure, surprisingly, a second inhibitor occupies here another transient hydrophilic cavity, this time, next on the other side of the active site. So conclusively here, the high pressure was able to reveal secondary binding sites, which are of the substrates, which are of lower stability. There is a growing interest for studying protein that can process molecular gases that are pollutant or greenhouse gases, like in first instance, carbon dioxide. But carbon dioxide is difficult to process in cryogenic cell because it is rapidly transformed into carbonic ice. So we are currently building here a new type of cell with two stages to avoid this CO2 icing, in which the crystal is pressurized in in carbon dioxide to produce the derivative, and is freeze into a neutral gases to avoid this icing. There is already more than 100 protein containing CO2 in the PDB, and the first example of use could be protein that can, or enzyme that can convert or capture or reduce CO2, like this one. Concerning general information, you can learn more about our high pressure freezing service by visiting our website at this address. And here on the right side, this diagram shows indicates the process that you can follow in order to benefit from the service of the high pressure freezing laboratory. If you have a high pressure freezing project, it is recommended to indicate this project in your beamline beam time proposal. But anyway, 15 days before the diffraction beam time, it is still possible. You can still send us all the information in order to have the safety approval to freeze your sample 1 or 2 days before the diffraction experiment. So the last slide to remind the paper that you can read if you are interested in our methods. I would like to thank people involved in the technical support of the lab, and here's a list of people that you can contact if you have a high pressure freezing project. And I thank you very much for your attention.
Thank you very much, Philippe, and thank you also for having stepped in to the sake of time. And now we'll move on to Antoine if he's back on a different computer.
Is it working this time?
Yes, yes, we can see it.
Okay. I'm really sorry. It's the first time I've seen this problem and I don't understand what happened. Okay.
Sometimes it's just the firewall. Sometimes it happens also in other cases with firewall, which are too. And here again, please, if you go into the display settings on the top left menu, because we see your notes.
Okay.
Perfect. Thank you very much.
Okay. Good morning, everyone. So I'm here to present you the ICOS lab of the latest developments. So as always, I explain what the ICOS lab can bring as information through complementary spectroscopy techniques. So it's an IBS ESRF platform, which is located at the ESRF. And it allows you to do various types of optical spectroscopy directly on crystals at cryogenic or room temperature. So here at the bottom, you see the location of the light beams, which coincides in the middle at the location of the crystals. And so you can do absorption by using 2 objectives focusing light on the crystals, fluorescence by using a 3rd objective, and finally a 4th objective in backscattering mode allows you to do Raman spectroscopy. And so you can get very nice absorption spectra from thin crystals like that one. And so this is useful because many proteins are colored. There are proteins with metal centers. So in this case, various redox state of cobalt. Proteins have cofactors such as flavins and chlorophylls. There are proteins whose function rely on the absorption of light by chromophores, here retinal, but also biliverdin. And finally, it's very convenient to understand the properties of fluorescent proteins. So why do you want to do optical spectroscopy on crystals? It can be used in complement to crystallography, first to determine the functional state of the protein in the crystal when it comes to interpreting the structure. At cyclotrons, it's very important. It allows you sometimes to evaluate the extent of specific radiation damage to exactly the groups that absorb light. And finally, because proteins are most often active in the crystalline state because there's a lot of water in the channels, you can do kinetic crystallography experiments, which amounts to determine the structure of unstable species in time of dose. You can do this characterization before or after the diffraction experiment. We call that the offline setup, or during the diffraction experiment, and for that we have developed several online setups. The offline setup is now in between the new ID29 and ID30B in the Chartres hall. You have here the control cabin and here's the experimental hutch. The latest development these past years have been the automation of crystal centering and the precise positioning of objectives. And it's been done with standard beamline equipment, a mini diffractometer, backlight, front light, and precise translation stages. Before going to the online setup, I guess it's a moment to say a couple of words on the FIB2, which is the new French CRG MX beamline, which replaces BM30A FIB. It has opened 4 months ago with brand new control cabins, experimental hutches, and optical hutches. The detector is now a Pilatus 26M. The sample changer is still a G-Rob with Texonic spine pucks, and we'll be able to put crystallization plates in the near future, and the remote mode will be possible by the end of the year. The top height beam capacity of FIB has been maintained, so you can adjust a rectangular shaped beam from 50 × 50 to almost 300 × 300. It's a tunable beamline. There has been almost 10-fold increase in flux after the EBS, and now we are in the range of 10 to the power 12 photons per second. And now the typical data collection on FIB is 3 minutes, so quite an improvement. So I mentioned FIB because this is the first beamline on which we can put the ESRF microspec for online UV spectroscopy that is suited for large crystals above 50 to 100 micron. And the beamline, the beam profile of the beamline is very nice in complement to the X-ray beams of other structural biology beamlines, which are now thin and Gaussian. So for radiation damage studies where you want to simplify as much as possible the factors that come into play, it's convenient to have this rectangular profile. Igor already mentioned the new development in terms of online microspec. So on Massive 3, very soon you'll be able to routinely record spectrum from small crystals, and I'd say in the 20 micron range. And this has been achieved by using off-axis parabolic mirror. And you see that it's very compact and fits well the experimental setup of Massive 3, and it will be suited for small Gaussian beam. And finally, the online Raman spectroscopy modality, which had been originally developed on ID29, had to be developed on another beamline because ID29 has now a very, very small beam. And this year, we should be able to propose this on ID30B. So this type of experiments will be very well suited for subtle studies where X-rays break specific bonds. And there are quite a few examples of this. And I will finish by our latest development, which comes as a close collaboration with the ID29 project of time-resolved cellular crystallography beamline. So on ICOS, we have developed a time-resolved in crystallo-optical spectroscopy to do pump probe experiments, where the pump is a nanosecond pulse and the probe a xenon flash time. And with this, you'll be able to record transient absorption spectra on a microsecond to 10 microsecond timescale. But I will leave Daniele to present the setup and the first application. So in summary, you can do all these various spectroscopies offline already on ICOS. And very soon, you'll be able to do UV absorption on FIB2, also on Massive 3, and the ID30B Raman this year. To apply for experiments, you just need to drop me an email or a phone call. Here, these are recent ICOS highlights from the past years, which illustrate the various applications. So on this example, you can monitor the loss of fluorescence upon the mechanism of a protein. Here you can characterize the ground state and intermediate state of important reactions by UV absorption. Here you can identify the complex conformational changes of a ligand. In this case, it's a fluorescent chromophore, but it can amount to a chromophore or a ligand. Here also characterizing the starting material for a time-resolved diffraction experiment. And here also perform at cryogenic temperature. And to be able to develop and support these instruments, I have to acknowledge many people from ESRF and IBS and also EMBL. Thank you very much.
Thank you very much for this great news and also the implementation in 3 beamlines for doing all this in cristallo optical spectroscopy of various type. We have time for a few questions to all of the panelists. I would like to call all the panelists here. You can either raise your hand or write your question in the chat.
I was wondering regarding the SAX beamline BM29. As you know, we are very keen on coming to visit it and we'll be there in a week's time. What our problem is usually that we are coming to with too many people, and since there has not been any back training lately, we are only allowed 3. So is there any plans on soon providing some back training again, or can there be some special training for the BM29 so that we can again send 4 people for free?
We will train everyone that comes. The 3 people rather than 4 is more of a, just the user office was instigated that, that's not down to us. But we probably should have a back training session for the Beamline now. We've changed a lot of software, so we will.
Yeah, so the users are very experienced, so it's not that we really need the training, but it would be very good to get the 4th person for free because it's not cheap to send them from Norway.
Yeah, well, I'll speak to the user office. I think that's we'll see what they can see.
There's one question to Philippe from Pedro Matias. He's asking who is the supplier for the glove box that you have installed.
I don't. Can you hear me?
Yes.
Don't remember actually, but I can check and answer him later. It's an English company, but the glove bag, it's not a glove bag, it's a glove bag, but it's a very simple one. You cannot go below 100 ppm of oxygen level, but I can answer him accurately later if I don't have the name in the head. No, sorry for that.
Next question for Sasha and Andrew. Is it possible to send the crystals to ID23-1 and ID30B for automated data collection? From Professor Nair.
Hope yes.
It should be possible in the near future, I think. Maybe Christoph can comment on it.
Again, it's something which we're working on, and we're trying to make this one available as soon as we can, as we have debugged it to a maximum. And then we will send an email around to all of our users and ask them who wants to be the first one to come.
Yeah, it's the same situation from 23.1. Yeah.
Thank you very much. From Tomika Jander to the SACS, can the user download all the data for the chromatography runs?
With the new HPLC into the BSX cube, that should be saved in that data should be saved into the H5 files. The older data is kept in the UV data files directly from the HPLC. They will be in the folders which can be downloaded. The text files should have that data in them. If they have troubles, they can contact us and we can use our proprietary programs to fetch that data out for them. But it should be in the new software, that should all be recorded and deposited in the H5 file.
And the next question is it possible when you're in remote, I suppose, to run a BSA reference on the HPLC before doing the actual run?
We can, it just takes time. So if you want to run BSA on the HPLC, we can definitely do that. Yeah. You just need to ask that we do that.
Now we have three talks from other upgrades, and we will start with Dr. Anton Popov, who will tell us more about the microfluidics for macromolecular crystallography and SACS. Up to you, the floor.
Thank you. Can you hear me?
Yes.
Good. So then, I'm still not a co-host, so.
Try to see, otherwise I will ask Lina. Try to share, or is it blocking you?
Can I? Is it?
Yes.
Oh,
yes,
I'm sharing.
It's coming.
Okay,
perfect.
Great. So, okay, then let's roll in. So my name is Anton Popov, and I will tell you what we are doing here now at the structure biology group with the microfluidics. So here you can see, let's say, kind of a typical microfluidic device, which is 3D printed. We are using here 2 3D printers, which are on the base of digital light projection, and we are using 37 and 27 micron pixel sizes. So the smallest printable structure of a good quality is about 2 pixels, so you can estimate that it's about 60 or like 70, 80 microns what we have. And the layer thickness, because it's printing in the layers, is from 15 to 25 microns. All chips which are printed on the methacrylate family-based resins. So this is kind of device is made for screening. So you're creating the droplets with different conditions, crystallization conditions, because you have 3 inlets for protein, buffer, and precipitant. Then you have this 3-in-1 mixer, and then you have a storage serpentine. So here you can see actually how it's like the process of mixing. With a 3D printer, it's possible to build actually complex systems, because you're working not only in 2D, but in 3D, like in all 3 dimensions. So you can actually create reservoirs, basins, different channels, like inlets and outlets. We are using, before, this is kind of microfluidics version 1.0. We've been using micronit holder and micronit based home printed chips, and also homemade devices like this. All of the devices were created by me and my dearest colleague Peter van der Linden from PSCM. So here you can see actually how the mixing is happening. So you can see the blue buffer protein and precipitant, and such nice droplets are getting inside the chips, inside the chip. So actually here you can see the result of this kind of mixing. You can see this is inside one chip. We've been playing with the gradients. So through this formation of microbatch droplets, we managed to show the gradient of crystallization. So this was the trypsin from bovine pancreas. This was our test protein, and we could show that within one chip you can see really gradient, and then you can actually choose which are the best ratios of buffer, precipitant, and protein. So this is actually what we are using also now. This is so-called Microfluidics 2.0. These are already also home-based devices, and they are all kind of beamline friendly, because this kind of goniometer holders for microfluidic device that you can see on the left of the screen is a two-part unit, also 3D printed, with 1/16 inch inlet connectors, and you put the chip inside, you put on the connectors, and you can actually run the flow experiments directly on the beam. And or we have also another kind of storage serpentines, which I showed just before, to hit the crystals through the chips as well. I will explain it a bit later, but this is just you can see just that we are actually eager to work together on different topics, and then we have actually a lot of stuff going on the beamlines already now. So the usual dimensions for the chips we are using are this, like the small ones is 16 to 8 millimeters and 1.2 millimeter thick, and the big ones are, let's say, 35 to 25 millimeters. This is the same size which I was using on MX beamlines and which we are using in the sample exposure unit for the SACs. So here you can actually see how it's happening. I made this GIF animation. So this is the holder. This is the chip which goes inside, and you use just the fitting to tighten it. So, and actually now you can just flow through the chip, and you have this kind of, you can see that the chip is not falling out, and this is kind of actually goniometer, so like with the magnet, so you can see that it, yeah, and it's placed on the beam. So we have released a really nice paper last year, accurate and rapid 3D printing of microfluidic devices, which shows actually how it's working. So all of the devices we are working with are kind of assemblies where you can change particle functional, practical functional parts like mixers or like this serpentine storage for the droplets, and just print it out and carry out a special experiment, which usually I needed on the beamlines, so, or like in the laboratory environment. So if you want to test something, it's actually quite fast from the idea to realization. So you can see here that it's different elements. Then you can see variety of different mixers we can actually print, and we are printing and we are using. Then you can see crystals which are growing inside the chips, and in-chip diffraction, which is really important because it's literally in-chip diffraction. So the next project, Microfluidic Project, which is carried on by the structural group, is a Peltier-based experimental device, so-called Thermobox, to investigate the thermal-dependent protein and the crystallization kinetics, like the growth of crystals. So this is the aluminium table inside the PMMA box with two Peltier elements, and which can carry out the gradient from -10 to 50 degrees. And you can store in this kind of environment. Actually, if you need, you can also pour gas inside or something to prevent the, like maybe evaporation or condensation. So I've been using these droplet microfluidic chips which you can see on the picture, which are already on this work stage. And I've been working with five different proteins, test proteins, to show that it's working and to show that it's possible actually to find first the ideal criteria for getting crystals, and then to grow as much as possible crystals, or as big as you need crystals. So you can see here on the left the microfluidic device, which is also in the unified goniometer holder. And then here you can see that it was lysozyme, trypsin, taumatin, insulin, and thermolysin crystallized already in the microfluidic devices, and they were brought to our beamlines. Here you can see actually this chip in the goniometer holder on ID30B, and there were really nice crystals of insulin inside, and obviously there was actually this mesh scan picture can show you it's exactly the same place which you can see, and you can see that the diffraction signal is quite dense, and there are a lot of crystals. And we could get with Christoph the resolution about 1.8 to 2.5 angstrom direct from chip, which is actually quite a lot. You can see this halo, which is actually the halo of material, but you can see that there are a lot of diffractional peaks. So the next thing is the sample exposure unit, which was today mentioned by Mark, which will be the third option on BM29, besides the robot, like BioSax robot, the sample changer, and the HPLC system. So actually, this is a vacuum camera with XYZ stage, and using syringe pumps, there are like 3 inlets and 1 outlet. You can carry different experiments inside the camera, inside the chamber, and with the camera you can actually follow what's happening inside. So, and actually this chamber has really big potential. So here you can see first test devices which we've been using there to test. We've printed unified holder for the sample exposure unit. Then it was a block device with already connectors inside. You can see it here, the connectors which are actually connected to the chip, and with the tubings which are leading outside the chamber. So for the windows, for the first test were used the silicon nitrate windowses, 1000 nanometer, 1 micron thick, and the COC film. I've been using the Permatex silicon glass sealer, which forms a tough, waterproof, and durable connection with the chip and the window. And you can see here how it's going. It takes around 40. It was taking around 40 seconds, like a pit stop for the F1 to change the microfluidic device into the sample exposure unit. And we've changed a bit this kind of fast door. Now it's really fast to change, but there was one weak point, and I will explain it also a bit. We'll show it a bit later. So with non-window microfluidic device, just the fittings were holding up to 10 to -5 millibar. And here you can see that I was using already the syringes, and I was flowing direct through the chip with window, at a very high flow rate, which is usually not used in the microfluidics, 25 microliters per second. And you can see that it's not losing water. which is flowed, and it's still 10 to minus 3 millibars, 1.4 10 to minus 3, actually almost actually higher that we have on the beamline. So this is 2.0, again, like microfluidic for SAX 2.0, which has a really nice unified holder where always fittings are already connected and you need just to change the chip. So you don't need to screw and unscrew all the time the fittings. And so you don't need to kind of break the vacuum of the holder, and not to over tighten or over screw and turn up the fittings. So here you can see that it's like, again, unified design, and you can change just the insides, print out the chip and carry out the experiment within 1 day. So in general, the idea was that maybe our users will have kind of ideas in their heads. They are telling us what they want. We are making the chip, and they can, when they are here, they can actually run kind of special experiment. So here you can see the holder and the common mix chip, 2-in-1 mixer. And this is how it looks on the, actually on the beamline. So inside the chamber. So you have, again, kind of platform, holder, tubings, and the chip, which can be changed just also within like 20 seconds. And the work vacuum was 4.5 10 to 3 millibar, which is again higher that we actually need on the beamline. Here you can see some curves that we've tested already, different materials. This is the COC film, silicon nitride, and Kapton film. And we have already done some flow experiments that with Petra, and we could see on the curves the changing between like empty microfluidic chip with water, then we flowed the buffer, and then it was protein. It was actually like the BSA, so we could see the differences. And also the good stuff is that there are some gels which are used by our collaborators, and we have run gels, what you can see on the right part of the screen. And it was just put inside the special chip and just used the Kapton tape to close it, and it was holding the vacuum perfectly, and we could have a really nice results. But about this, I think there will be another presentation, or this colleague will have also a poster. So the second thing for the chamber was the high throughput microfluidic cell, which is already printed. It's also kind of cell where you can you are closing one part of the chip with a Kapton foil or with any kind of window material you want. You pipette samples, or if it's gel, you just spread it across this kind of 4x4 square, and then you make another window, and then you just, again, you can see that's the same size and the same fashion. So you just put it on the holder, which is already there, and you can run the experiments. So this is also 25 to 35 millimeters to fit on XYZ stage. This is laminated sandwich, like with the Kapton film, which is 12 micron, and the well volumes are just about 1 microliter. In this kind of things, they are reusable or disposable, because actually the price of this kind of chip is quite low, so we don't care if you have some special, I don't know, requests. So it can be just reused or disposed. And just to have a summary about the sample exposure unit. So we have unified holder, and we already have a high throughput microfluidic cell, flow cell, just with one window. This is kind of a brother of a capillary, what we're using now. And we have the mixing cell. So if you want to see like the real-time changes, it's also possible to run already these kind of experiments. So actually, if you are ready for collaboration, or if you have a lot of different ideas, we can just talk about them, because actually sample exposure unit can be already, at least in the experimental way, can already be used by our users. And the last part, which I will just briefly talk about, it's also serial crystallography microfluidics, which is carried out also on our MX beamlines. This is kind of also 3D printed injection system, where the crystals were just pushed through the COC film U-turn. And this kind of experiments were carried out on Massive 3 and ID30B as well, and at ID23-2. This also, everything is home printed. The work channels were around 150 and 200 microns, and the sizes of, you could see the diffraction peaks, and the sizes of crystals were about 20 microns. What also have been already tested on ID23-2 with our other collaborators. I think maybe Daniele De Sanctis will tell us also a bit about this experiment. So this is a 3D printed microfluidic flow cell. You can again just flow the sample or flow the small crystals, or you can mix, add something if you want. So it's also placed at a like homemade and unified goniometer holder. So that means that you can use it actually on all of our beamlines. And window thickness, it was this diffraction signal, we got it from the polymer window, which is actually the material of the chip. And the thickness of this chip, of this window material was 350 microns before the crystals and 350 microns after the crystals, after the samples. So even with the window, with this not preferable one actually material for the window, we still could get the diffraction signals with the exposure time of 30 milliseconds. So we have observed already even like these designs. And on the right part of the screen, you can see exactly this kind of unified holder, which I was showing on the GIF animation just a bit before. We've been also talking with Daniele about serial crystallography, and with Julia actually about serial crystallography, which will be carried out at ID29. So we've been discussing this old paper of 2015 of our colleagues from XFEL, and I was already printing and doing some kind of this grid to sit to have the crystals spread on them by the holding by the capillary effect, which is actually a bit hard to print out with our 3D printers. So we decided to make this kind of traps, and as I was working on them already before with Peter, this kind of, you can see here the nice lysozyme crystals in kind of park spots. So I've been already creating this kind of design, like designed and printed out already kind of this trap system. I just redesigned it a bit to have more crystals inside, and you can see the idea is to flow this kind of solution where the small crystals are there, and they are sitting in this kind of bottleneck, and then you can just shoot through them, as we have shown before that we can actually shoot through the chip. So I think it's a really high possibility to carry out this kind of experiments. So last part was also just droplet-based thing to work with membrane proteins, just to maybe encapsulate or just to work with LCP. Last year, I already carried out some experiments with the lipidic cubic phase, and which is quite dense to use with the microfluidic, but I could show that it's possible to work with that. So we've been also deciding with my colleague Julien to run kind of to restart again these kind of experiments with the lipidic cubic phase and with the membrane proteins. So actually, you can see that there are a lot of going on with the microfluidics. And if you have any kind of questions, you should contact me or Peter van der Linden from PSCM, and we will try as much as possible to make you happy with the microfluidics at the structural biology group as well. And thank you very much for your attention. And I want to thank all of my colleagues with whom I'm working. So,
thank you very much.
Thank you very much,
Anton. We have time for just one quick question. Otherwise, we will do the question at the end. There will be a time for roundtable at the end. Any quick question? It seems not. So thank you again, Anton. And we will move to Dr. Serena Rocchio.
Yeah.
Who will present.
I stopped.
Oh, sorry.
Yeah. No, I stopped. Is it okay?
Yes,
Yeah.
Yes,
okay,
it's okay. It's okay.
Thank you.
About Crystal Direct at Massive 1. Serena.
So yeah, can you see my slide?
Yes.
Fine? Okay, yes. Perfect. So today I'm going to show you the latest update from Massive 1. And in particular, I want to introduce you this project that we are working on in collaboration between ESRF and FBL that involved integration of the Crystal Direct harvester in the beamline environment. Before doing so, let's have a brief update on the beamline. So Massive 1 is a unique facility for high throughput, fully automated data collection of macromolecular crystal operating without user intervention. The data collection is optimized for every single sample where the user can interact with it by providing information in the diffraction plan in SPB. We operate through a flexible booking, so the sample enter in a queue system, and it's a fixed energy beamline with an high intense beam and a flexible beam size, so we can go down to 10 micron. We do have an MD2 as diffractometer, and the beamline has been equipped with a high capacity QR. We have a Pilatus 2M, and we recently introduced the Crystal Direct harvester. So at Massive, we automatically collect samples that have been harvested on a variety of sample holder, and to do so, we run a loop centering and an Excel centering in order to locate the best diffraction part of the crystal in the center of the beam. We can also collect different datasets from the same beam when there are multiple samples if it is indicated in the diffraction plan. Moreover, we can also do a pseudo-helical data collection in which we collect multiple partial datasets from different positions within the crystal. So the data collection is automated, but it's the user driving the parameter usage. So in the diffraction plan, there are two and three that are mandatory, so the protein acronym and the sample name. And if no other information is provided, we run a general workflow that allows us to detect the best parameter for your crystal. But the user can interact with it by providing information on the experimental type, choosing between different workflow, or fixing some parameter, for example, the resolution or with the beam size or the total rotation angle. And this afternoon there will be a tutorial on workflow in Massive 1. So if you want to have more information, that's the place to be. Just to summarize how we do collect data. So we load the sample, we run a mesh scan in order to locate the crystal within the beam. We run a line scan not to miss the crystal in vertical direction, and we do a characterization on 4 images, and we follow the advanced strategy to collect the data. The data are processed through different pipelines, and if you provide also a model or a CIF file, you can also have structural solution ligand fitting. This is the general way in which we do operate. We are also working to improve the data collection of low symmetry space group. We have a collaboration with Global Phasing in order to integrate their workflow in our pipeline. For the moment, we are taking advantage of the mini-cap agoniometer head that you can see here in open and closed form formation. And therefore, in case we detect a P1 space group, given that it's a really low symmetry space group in which a small amount of data can be lost in blind region, what we do after the first data collection, we open the kappa and we collect a second data collection, a second data set at a different crystal orientation. just to give you an example of what we can do with this workflow, that was a pilot project with a space group P1. In this case, more than 100 datasets were analyzed in order to define the binding pose a specific region. But as you can see here, the density for this compound was quite poor by using a standard protocol. But applying this workflow, we were able to improve the density and in the end to fit the compound in within the density. In this context, it's pretty clear that now the automation is changing the way in which we are processing and analyzing data, and but is also seeking the ground to work on different pipelines to respond to dynamic changes on scientific needs. So in this context, what we decide to do is to integrate the crystal direct harvester in the beamline environment. In particular, what we want to do is to be able to harvest the crystal and directly load it to the beamline. To give you a bit of a context, if you are not familiar with it, the harvester has been developed at EMBLI in collaboration between the markets team and the instrumentation team that allow access to a fully automated crystallography pipeline that is controlled through the CRIMS interface. It is the core of the crystallization facility at EMBL that offer different kinds of services from protein crystallization to structural solution, fragment screening, and serial crystallography for membrane protein. This technology is based on the fact that the crystals are grown on an ultra-thin, low-diffracting film. The crystal is excised through laser photoablation and then placed on a sample holder. In the EMBL facility, the crystal direct operates in a cryo mode, so the crystals are harvested and then stored in a large QR, and after that, they can be shipped to the beamline. So here we want to combine the harvester with the automation on the beamline in order to explore different plate-to-beam application. So here we are. This is how the arch looks like at the moment, a bit crowded. So we have the harvester, the flex with the sample charger, and the MD here. And all this installation was possible thanks to a really good collaboration between the ESRF staff and the EMBL instrumentation team that make this transition really smooth, and they were quite responsive during every phase of the installation and cabling up to the operational mode. I'd zoom in just to show how the harvester look like inside. Here you can see the plate that has been loaded, and here there is the sample ready to be loaded on Gonio. On Gonio, we have the possibility to swap between a cryo stream and a humidity device control. So just as a reminder, what we want to do here is that we want to harvest the crystal and mount directly them to the beam. To have this process happening in an automated mode, we have to provide to the harvester a list of samples that we want to use in our experiment. So we can do so in the CRIMS interface, in which we can prepare an harvesting plan. So we can choose the crystal that we want to use in our experiment. For example, here, we want to harvest this crystal using this specific cut shape, but it's also possible to choose multiple crystals as this one here. So once we have the list of samples for our experiment, we send that to the harvester that will executing the harvesting action. So this is how the software look like. So I want you just to focus on the central peak because to show you the process. So this is the drop with the crystal that we want to harvest. We aspirate the liquid around the crystal to cryoprotect it. There is the laser cut, and the sample is here on a shape ready to be loaded on Gonio. So to reach the full automation of this process, we had to integrate different software from CRIMS to MX-Cube to iSpyB. So the basic operational mode here is that we prepare the crystal in a crystal direct plate. When we are happy about our crystal, we prepare an harvesting plan. So we send the list of crystal to the harvester, which is the main actor here, executing the harvesting action, and this list is also sent to MX-Cube. From MX-Cube, we want to operate in the same way as for other samples. So we can retrieve the list of samples as a queue system, and we can assign them a workflow. In this case, when we start experiment, what happens is that the first crystal in the harvesting plan is harvested, mounted on Gonio, and we start the data collection. As soon as the data collection for the first sample starts, that action triggered harvesting also of the second crystal in the harvesting plan. Therefore, the harvesting action and the data collection are coordinating and are happening in a cycle up to the moment in which we reach the end of the list. So where we are at the moment, we are validating the different data collection modalities in which we are interested in. So we start with the basic use case, so data collection at cryo temperature. What we can do at the moment, we are able to harvest and collect data in automated mode without user intervention by using a specific workflow. And in this case, we want to work on different target-based applications that, like, for example, the systematic analysis of crystallization condition. We have a humidifier device control on the beamline, so we want to really take advantage of that, and we want to render the dehydration experiment more accessible to user and automated. It has been long known that the dehydration can improve the crystal diffraction quality. So we have a workflow with configurable parameters that we can use. And here we can really take advantage of the queue system in order to shorten the experimental setup, because we can queue up different samples and assign them different workflow in which we can test different parameters. And that will help us to not only to shorten the experimental setup, but also to make this experiment much more reliable and reproducible. In the end, we are really interested in room temperature data collection, which is kind of a topic at the moment, to look at structural data at physiological temperature, to study protein dynamics, ligand binding site, and protein velocity. So here we are working on a workflow in order to do a fast data collection with an attenuated flux and a small scan. So just to sum it up, we integrate the crystal directly in the Beamline environment, and that will allow us to expand the portfolio of automated services offered by Beamline, and will allow us to work on different plate-to-beam applications. We are focused on improve the efficiency for high throughput application like screening. We are really keen to render the dehydration experiment and room temperature experiment more accessible to user, and also work on some target-based experiment. To do so, we will keep working on room temperature and the workflow optimization in order to validate the system with multiple targets. And we are looking to work on room temperature, serial crystallography, and in situ data collection. We are aiming to open to user in mid-2022, but if you are interested in one of those applications, please be in touch with us. Finally, we will keep working with the instrumentational team in order to render the full process automated. What I mean is we would like to have a placeholder in order for the flex to load and unload the plate automatically, and also a pin cleaning station, so that the full process is completely automated. In the end, I want to thank all the people involved in this project, which are quite a lot. It's a collaboration between EMBL and ESRF, just in order. That's from Mccarthy team, in which Matthew Waller is the project manager, and markets team, with a strong collaboration with the instrumentational team led by Kirkley Pop, and from the ESRF side, the experimental division structural biology group, in particular Didier Lorizzo, which is managing the Beamline with Matthew. And if you are interested in this kind of application, I will have a poster tomorrow, and Nicholas as well, that is working on in situ data collection. And thank you for your attention.
Thank you very much, Serena, for these upgrades, and also for having been perfectly on time. Are there questions? I have one. If in your drop you have more than one crystal, how can you harvest more than one since you are dehydrating?
Okay, that's true. But we see that when we can collect data, we can harvest the first crystal, collect a data set, and also perform a second data set on a following crystal, because as soon as the data collection starts, the following crystal is harvested and this is placed in cryo position. So the crystal is harvested and this is waiting under cryo. So it's cryo protect.
Okay. Thank you very much. If there are no more questions, we can still go to the next speaker, and then we will resume with all the speakers at 12 at the end for a panel. Now, Dr. Daniel De Santis for the news and views on ID29. Daniele.
Yeah, I'm ready to share my screen. Okay, can you see the screen clearly?
The floor is yours.
Thank you. So I'll give you an update on the progress of the installation of the new ID29, also in provision of the opening for users, opening for friendly users, and more importantly, for the deadline for the beam timer application that will be on the 1st of March. So just a reminder about the project. ID29 is one of the flagship beamlines that has been designed to fully profit from the new ESRF EBS source, and has been designed in order to be able to get a very hot beam, so very high flux density, in order to have a very short exposure time on the crystal. So this is the changes that were given thanks to the new machine with the current source. While in the now it's getting near future, we will update the U21 ondulator that you see here in blue dots with the CPMU 16.2, which is here in green, which will yet brings an increase in the brilliancy of the beam at the sample position, and extend the range of energy operation that is provided just with a single ondulator. So the plan is in the long term to install it at the center of the straight section in order to reach the minimum gap and then the higher flux. So the new ID29 has been, the project has been proposed in order to bring silver crystallography in a very similar fashion that has been performed at FEL to the 4th generation synchrotron. Of course, it's not we're not going to be on the same kind of exposure time and pulse length as the free electron laser, but you will see that there is quite a certain interest to do this kind of experiment more frequently at the ESRF EBS. So for serial crystallography, you know that diffraction is occurred before the crystal is actually destroyed, and this has been a way, at least to a certain extent, to outrun radiation damage. Given the high brilliancy of the beam and the high focus of eye focusing, it's possible to store very small crystals of a few micron or even less, and to do that then at room temperature, so to avoid to have artifacts that are being induced either by the cryo temperature or by the cryoprotectant. But in this way, the diffraction is collected from still crystals because the exposure time is very fast, so there is no rotation on the crystal. And the new methods for integrating the data and process the data have been developed since then. In this way, we collect data from thousands of crystals and that are sitting in a random orientation, and combine the diffraction pattern from all these crystals. We can reconstruct the reciprocal space and eventually calculate the electron density map. And of course, this opened a new way to perform time resolve experiment. So as I said previously, the possibility of collecting data at room temperature permits not only to, as in this case, to reveal the thermal, different thermal conformation and thermal movement as they can be observed at room temperature versus cryo temperature, but also to outrun, to escape conformation that are energetically favorable at cryo temperature, that are not physiologically relevant, or that they can hinder the possibility to adopt conformation that in which the protein can bind or process a determined substrate, and also to avoid the presence of cryoprotectant that in many cases they can mimic the substrate. Think, for example, to the presence of glycerol for proteins that bind sugars. The fact of working with microcrystal at room temperature also opened the possibility to perform time resolved study because the proteins, the side chains can actually move, and since the protein at room temperature, then they can be activated or soaked. So in the case of activation, that would be pump and probe experiment, where we can activate a photosensible protein or a photosensible compound inside the protein, and then record the diffraction pattern immediately after, or with a certain time delay, and then reconstruct what is the structural evolution with the time. Or to direct mix, as in this example here, a solution with nanocrystals with the substrate, a compound, or even a different buffer to induce pH changes, pH-dependent changes. And as I mentioned before, this opened the possibility to do, for this kind of experiment, to do time resolved study, either by pump and probe or by mixing. With third generation synchrotron, this kind of experiment were pretty much limited to lower experiment. So with pink beam, so very large bandwidth, and performing a single pulse experiment in the timing mode of the storage ring. The fact, of course, that there was a problem of radiation damage because the crystal was a large crystal, the beam was quite large, and the crystal were heating. While serial crystallography done at the new extremely brilliant source permits to overcome most of these limitations. Of course, to do that, we had to, and you got some example also before in Anton talk, to bring some of the sample delivery methods that were developed for serial crystallography of free electron laser to the synchrotron. And here you have some examples of high viscosity extruder, microfluidic, as it was presented by Anton a little while ago. And but there are also new methods that are in continuous development, such as the possibility of using tape. And here you see a schematic representation in which a crystal slurry is deposited like a fountain pen on a tape, which is continuously unrolling, and then diffraction images are recorded as soon as the crystal is in front of it, is in front of the beam. Another quite effective way is to use a fixed target, and by using this silicon chip that they present, unlike other kind of devices that I showed you before, a pre-location of the crystals, because the crystals are going to sit in a well-ordered array, and in this way we are going to collect diffraction image in from each of the crystal, which is located in one of each hole, which is which, as I said, it's a regular array defined in this compartment, which is part of this larger support that makes 3 centimeter by 3 centimeter. So in this way, you will see a little movie in a few slides. We collect diffraction data from each of the crystal which is sitting there. So this maximize or better minimize the sample consumption and maximize the hit rate. So in some certain cases, so for low amount of samples, that could be more convenient than using, for example, an injector. So for the beam characteristic that we are going to have on ID29 compared to what was possible to third generation synchrotron, as I mentioned before, is the possibility to reduce the exposure time by increasing basically the flux density at the sample. So with the fourth generation synchrotron with the 1029, we should get an increase of almost a 5 to 6 order of magnitude in flux density, which brings us to exposure time as short as a few microseconds. In this way, it gives us the possibility to exploit the micro to millisecond time range for time-dependent reaction, which is very convenient because this is basically the time. It's the speed of the vast majority of the enzyme that we might be interested in study. So, in a sort of nutshell, ID29 is a beeline for serial and time resolved crystallography, which aims to be able to study photo activable process that either be in photoactivable proteins or by the presence of a photoactivable compound like H compound inside the protein, to study enzymatic reaction directly in the crystal, and also to be new tools for doing drug design because of the possibility to perform direct mixing of the crystal slurry just right before the sample diffraction experiment. So in order to do that, as I said, we developed a room temperature beamline for room temperature with a very high flux, and I stress once again for exposure time in the microsecond time range with a very high repetition rate. Very high repetition rate, of course, has a double advantage of minimizing the sample consumption for when it comes to injector experiment, and to maximize the data output and the efficiency of the beamline. Unlike free electron laser beamlines, it's much easier to tune over quite large energy range, and we had to develop a very accurate control timing system in order to trigger the events that are time dependent. And since, as you've seen, there are quite a certain variety of sample environments that are currently used, and we probably currently will use also for the few years to come for doing silver crystallography experiment. We have a sample environment which is rather flexible, and in order to facilitate the sample preparation before the experiment or a bit ahead of the experiment, the beamline is equipped with a sample preparation library where the users, well, you can come either to prepare the sample or to make some tests to evaluate the quality of the sample before doing an experiment. So unfortunately, I mean, even though everything of this is built, we cannot take a picture that shows everything. So I still have to stick to SolidWorks rendering. So this is the complete layout of the beamline. You have the two optic catches, the first with the monochromator, the second with the choppers that they make the timing system, two experimental latch that are downstream next to ICOS that Antoine presented before, and a large control cabin with the sample preparation laboratory, which is in the short rows, which are in the short rows all. So this is again just a schematic of the optic catch, the first optic catch and the second optic catch. So in the first optic catch, as I said, is the home of the monochromator. We have developed a new type of monochromator, which provides larger bandwidth. So we have, as you see, this is a double mirror system, and you can see probably in this image here, in this picture here, 3 different stripes. So these 3 different stripes correspond to different bandwidth and different energy range. In this way, we are going to achieve a bandwidth of 0.3 or 1%. This compared to normal MX beamline, you are at for normal MX beamline, you are at the around 10 to the minus 4. So it's a quite, let's say, more colored beam compared to monochromatic and classic monochromatic MX experiment. But this is actually being chosen for 2 reasons. The first one is to increase the flux, so to minimize the exposure time, and to thicken the UL sphere, as you see in this representation here, to basically compensate for the lack of rocking during the data collection experiment. In the second RTH, there are two choppers. One that absorb all the heat, because when we have 10 to the 15, 10 to the 16 photons per second, the beam is quite hot. So this is the first chopper that takes all the heating. And then we have a second chopper that we just recently received, that we have developed with the help of this Swiss company Celeraton, that I just showed that is one of the sponsors that was very for the user meeting. I'm very pleased of that. That developed very fast spinning motor with the magnetic levitating system. And they have developed specifically for us a special design of it that you see that it has a certain opening that we are going to use to make the exposure time, so to chop the beam, and that can run synchronously with the experimental clock that you will see in a second. The other very important integration is the Jungfrau detector. So the Jungfrau detector is a detector that's been initially developed for Swissfell data collection. But we are gonna basically tweak it, or basically push it to its maximum in terms of repetition rate. And with the Lima working group and the Beamline control unit, they are developing the new software, which is basically in sort of test version at the moment, that does the automatic reduction and is able to cope with the terrific data output that it produces, because each image is 4 megabytes, but the maximum speed that we can work here is, as you see here, 2 kilohertz. So this corresponds to 16 gigabytes per second that are spit out by the detector. And as you see here in the back, basically each model has an optic fiber connection in order to be able to cope with that. So once we get the data out from each models, we need to reconstruct the geometry of the image on the fly. And with the help of Nicolas Coquel, we are integrating, and this is a test that has been done either with the Eiger and for the Pilatus for the moment, to make a direct hit detection, so identify immediately which frames they contains diffraction by using the nanopixel software. So as I mentioned before, one of the most important development that we've been achieving is that we have been doing has been the timing system. So in terms of timing, we are going to synchronize with the machine clock. So it means that if you see, this is the storage ring representation with different electron bunch that they go around. We synchronize with these bunches here by using a CT module, which is a newly developed card at the ESRF to synchronize time resolved experiment. And we run the 2 choppers synchronously with that, and the same signal is sent to the SSX box, which is an additional timing device that is interfacing with the diffractometer and with the Jungfrau, and is able to activate or to send activation signal to some probe, which could be a laser, that could be an injector system, that could be anything you want, because it's just an electronic signal that is sent out. And the most important part of this is that then we can offset this signal with accuracy of 20 nanoseconds. So basically we can change the delay between a laser and X-ray pulse in steps of 20 nanosecond across 1 millisecond, which is the repetition time, which of course we can degrade, so going slower. So what you see here is the diffractometer that has been developed in a collaboration with the EMBL, which is this fast table, which is synchronized with the SXS box and the storage ring clock. And what you see here is one of these silicon chip, as you saw before, for the hair experiment. And we move each of these holes in front of the beam synchronously with the clock, which is provided externally. So it means that every time that we have X-ray beam going through the choppers, we are going to have the next hole of this just in front of it in order to collect diffraction, to record diffraction. And at the same time, of course, a trigger is sent to the detector to acquire the image. So we are currently finalizing the installation of the experimental latch that you see here. That in this representation, it also integrates another development that we are doing in order to facilitate serial crystallography experiment and to maximize the throughput of the beamline, which is what we call the Flex SSX, which is a sample changer that you see here, that instead of taking cryo-cooled samples, it takes this kind of support, or a slightly different design of that are stored in humidity control cassette that you have here. So the robot goes there, take one of those, put it on the goniometer, and then the collection can be performed. This is also to maximize the beamline throughput, as I said, because since we are going to go up to a repetition rate of about 1 kilohertz, and we have no limitation at the moment to go up to 2 kilohertz, it means that one of those chips can be screened in a matter of a minute or two. And this is a picture of a few days ago. So you see here, this is the KB system, the table with the diffractometer that is going to be entered next week. And we're still going to be a bit delay with this motorized support for the detector, but we have an alternative mounting system, temporary mounting system that is going to, where the detector is going to be sitting here. I mentioned also the possibility to trigger light dependent process by using the laser. So to doing that, we are looking at two different ways. One is through the on-axis viewer, so to add any laser injection port and with an additional beam splitter here so that the laser gets through the axis viewer directly so that the laser sees the same point as the X-ray, or just from a side, as you see here, as in the case of having an injector. I apologize for this image, it's a bit pixeled, but we have purchased a laser that is going to be delivered in June. So it's a laser with a very high repetition rate. It's up to 500 hertz, so to match half of the repetition rate of the data acquisition, with a very high tunability range from 100 to 2000, with the possibility to using the OPPO signal 355, and he has just enough power to trigger the crystals. You've seen some of those examples also in Anton presentation of the user microfluidic. Also in the context of the SSX bag, we have been tested quite a few different number of sample delivery system. Most of them, or if not all of them, they will be possible to do it to be used on ID29, and so we are also in the context of initiating the operation of the lab. We are open to provide you with help and support to do this kind of experiment, and to define, at least initially, a sort of pipeline where we can pre-characterize the sample to see if they are good enough for an experiment, as you will see in a minute. Yeah. So as part of the ABS Light project, Antoine has quickly mentioned that before is also the possibility to do time resolve spectroscopy analysis on the crystals at the what was previously called the cryobench, so the ICOS. So this is the time resolve ICOS setup, where you can study, actually pump reaction directly into the crystal and measure transient spectral changes in the UV visible range directly on a crystal gallery. And so it's equipped with as well with the nanosecond laser at lower repetition rate, because of course doesn't need to do as fast as on ID29, but you see that the energy range is basically the same. So the wavelength range of the laser are basically the same. So this can be used as a preparatory experiment before to pre-characterize the sample and to define the time point that you would like to observe structurally, and then move to perform the experiment on ID29. And this is a first spectra that has been recorded a couple of weeks ago, if I remember correctly. And you see these are crystals that are quite small, because this is 20 micron. And this is how the spectral changes of flavin is changes after 150 microseconds and after 200 milliseconds. So a few words about the sample preparation laboratory. So this picture you see, so this is now the lab is getting rather crowded. This was a picture before we start installing a lot of things. So it's possible now to come to the lab to do pre-characterization of the samples, and also in particular to do pre-characterization of injector experiment, as you see here. So we have now, Juliana is working a lot on this, prepared a bench offline where with a microscope camera, where we can flow the samples and test if the crystals are okay with the jet. If the jet is okay, flows well. We have also a humidity controlled environment where the crystals can be, where the chips can be loaded, or where the microcrystal can be manipulated and preventing dehydration. So don't hesitate to contact us if you want to run some tests even now. I mean now. Oh, this duplicated. Sorry about. So application for beam time. As I said, the Beamline, we are opening to take a beam time application from the deadline of the 1st of March. So I'm gonna go a bit slower on that to be clear, and then you ask if you have any question on that. The beam time application is gonna work through a bag system, but not through the regular MX bag system. So we'll need to make a bag application specifically for AD29, as it happened for the cryo-EM, I think. If you apply for the beam time through a bag, first of all, there's no minimum size of the bag, so there is no minimum number of groups. The important is that the group is able to use all the shift that requests, as it happens for other bag. When we are going to schedule the experiment, we are planning at least initially to do very long experiment in order to overcome initial difficulties that we can get, especially on the sample preparation and on the sample flow, to allocate 18 shifts, so to have experiment running from Tuesday to Sundays. But there will be the possibility to have a shorter session for sample testing, and we are going to reimburse, I think, at least one user to come offline to do sample preparation or jet experiment. And since it's a bug application, there will be 2 deadlines per year for application, usually in March and in September. And of course, this is more indicated, or at least thought for users that are planning to use regularly AD29. While for curious users or users that have just one project that they think can benefit from serial crystallography, at least initially, there is the possibility to do rolling application for the AD29, which as well it works as a standard rolling application. It's open. The call is permanently open. You get the beamline scheduled in a short time. And in most of the cases, though, there will be basically a single experiment or just a few sessions, a couple of sessions, maybe one to test the samples and one to do the real experiment. And as we should be able to welcome friendly users from May, June, feel free to contact us. In terms of proposal, they could be both methodological and biological projects. So even groups that they develop sample delivery methods are very welcome to submit a bag. If you would like to make a bag, or you would like to us to identify which other group you can cluster to form a bag, we are very happy to help. So as I said, for the proposal now, for the AD29, for the bag and the rolling, you have to go to structural biology. But if you select bag, in addition to classic MX bag or BioSax and cryo-EM, now you have also the option for AD29 SSX and time resolve SSX. For what concerns the access to time resolve ICAS, this can be done in a similar fashion as it's done when you request the use of the facility, the other facility for ICAS. So it's enough to check the box in the A form, and then of course this can be done for A-form or for ID29, but it can also be done for A-form for other MX experiment if you want to come here and pre-characterize samples on TR ICOS. Well, I'm gonna skip that because, well, no, not gonna skip that. So just in a nutshell, in terms of beam characteristic, I said we're gonna have the D29 a very long energy range for a serial crystallography beamline with two possible bandwidth, and with a photon flux which is gonna be higher than 10 to the 15 photons per second. A beam size with a submicron, and with exposure time, which are defined, the fast chopper that are in the microsecond time regime, which are corresponds to 10, 20, and 30 microsecond according to different opening that we have in the chopper. And the initial repetition rate is 925 hertz, so slightly below 1 kilohertz, not 1 kilohertz, because 925 is an exact division of the frequency which is provided by the machine. But as I said, we don't have no more technical limitation for us to go at twice that speed. So a bit, as I mentioned, a bit of the timeline. Of course, as you can imagine, we got quite a bit perturbation due to the different national lockdowns and delays from suppliers in order to get everything ready. But we are getting there. We are currently finalizing the installation of the experimental Latch. But the good thing is that since we basically assembled and pre-test a lot of things offline, most of the things are ready to be installed. And we already had, when we did the radiation test of the experimental Latch, an initial estimation with the unfocused beam of the photon flux in the, let's say, very not optimized optical configuration, and we already hitting 10 to the 15 photon per second in the experimental Latch. The bug proposals is on the 1st of March. For rolling, there is no deadline because the call is permanently open. We are happy to welcome friendly users from the end of May, because there is a shutdown in May. So from the end of May, beginning of June, in order, and that we're all going to use that time to prepare for the user mode, which will resume in September. And just let me acknowledge all the people, or at least part of the people that I could remember to include in this slide, and happy to take any question.
Thank you very much, Daniele, for this terrific improvement of ID29. And now I would like to call all of the speakers of the first part and also of the second part, because we are open for a roundtable of discussion on all the upgrades and news novelties that have been done at the ESRF. If there are questions, Pedro Matias, you can open your microphone.
Hello, good morning. Can you hear me?
Yes.
Well, first of all, I would like to congratulate all the speakers for the impressive presentations and increase and the continuing efforts to develop the ESRF as a first in the world lab and facility for carrying out synchrotron research in structural biology. But my question actually is more like a comment, and maybe more directly addressed to either Christoph or Gordon. I'm not sure who's the right person to answer this, but it has to do with user support. So as you know, since the restart after the upgrade, we now only have 3 supported users and instead of 4, and which in the case, in the general case, it is not so much of a problem because we now all pretty much have gotten used to do remote data collection. But when you need to go to the ESRF for specific experiments like the ECOS or the HPMX, it is complicated because 2 people at least need to go to make good use of the time, and then there's only 1 doer left. So if they also want to take their crystals for another experiment on the dewar, it means that the beam time cannot be shared unless the other groups pay for their own dewars. So my suggestion would be that in those circumstances, the ESRF should pay for at least one more person, one more reimbursed user added to the list. I filled out an A form for an ECOS experiment from one of our back groups. I don't know if it's now the same thing for the HPMX, but maybe additional reimbursed users could be included in those forms. So this is what I would like to say.
Okay, Pedro, I think there's a case to be made for having more than 3 users for sort of special experiments, if you like. So the ICOS, HPMX, maybe even on the new ID29, and perhaps even for BM29 actually, although we need to discuss that a bit further in the group. But I'll talk to the user office and see what can be done. That's all I can promise, Pedro.
Well, that's good. Thank you very much.
And still on this point, what about the news that the Calypso for transnational access is not going to be renewed? So how users of ESRF will be affected?
As far as I'm aware, we have very little to do in terms of our users with Calypso or Calypso Plus. Mm-hmm. All users apply directly to the ESRF BTAP for beam time. They don't apply via Calypso. The ESRF is actually not eligible for transnational access in the sort of traditional sense. You can get it if you're an SEM, which is one of these SME, sorry, which is one of these small and medium-sized enterprises, I think. So for the structural biology group, I don't think it will make that much difference, to be perfectly honest, Adriana, but I also have to say that I don't really know that much about Calypso and Calypso Plus because we don't have that much to do with it.
Thank you. There's a question from Mark Raji. Great to hear about the development of ID29. There will be some overlap with the P14 of Petra or some friendly competition. So what do you think the advantages of ID29 and P14 will be? Will there be some specialization in each of the 2 beamlines, or will they go in parallel? Daniele.
So there is clearly an exchange. We have discussion both on the sample delivery part. I mean, there's no really competition. I don't think that personally, I think that competition in science it can often be misleading. No, there is an exchange. The fact that, of course, for the moment, I think there's a certain of complementarity. There will be more overlap when Petra 4 will come online, because then we are talking about similar sources and more comparable flux. So for the time being, ID29 is going to stay as the first beamline with this kind of characteristics. But of course, as I said, there is a common development, there is a common exchange. We are all developing to similar things. So does it answer your question?
I suppose then Mark, if you want, you can open your microphone. This is open. I have one question to Andrew Mccarthy about the plate reader of ID30B. Is Andrew there?
Yeah, I'm here.
Hello, hi. Is the reader specific or just one type of plate, or as long as it's X-ray clean, everything can go, or?
Yeah, so what we did at the moment is to configure it for 3 of the most common ones. So one being the Crystal Direct, which the HTX lab use, and it's also available from Metagen. And then there's the Crystal Quick X, again, that's available from Mitagen, that's Grainer. And then there's the in situ from Mitagen. So those 3 plates are tested and can work. And then we have some custom ones that we made for the microfluidic chips. So Christoph and one of the technicians came up with a design to hold these special microfluidics chips. So if you have a different type of plate, we can also have a look. There's a little problem with the thickness, so you just have to be careful that I think there's a special format, SBS 96 or something like that.
Thank
Does
you.
that answer the question?
Yes.
Yeah.
Yes. Well, already 3 of the commercial one, so it's a large choice anyway. It's not just one, which is really good. Are there questions from the audience? People are quite quiet. May I ask another question? This time to Antoine. Because he mentioned that on the beamline it can be done on FIP2, on Massif, and ID30B. So shall a person just need to specify what he would like to do in the form, in the application form, or it can be done also after the beamline has been already just on the A form? Don't know if my question was good.
Yeah, I'm not too sure. I'm not too sure of exactly the question, but what I recommend is to send me an email, or to tick the A form. Anyway, it will send email to the team. So I'm not sure of the precise question, but
yeah, the question was that you said that
complementary spectroscopy. Send me an email, and I will advise you how to proceed best.
Yes, because you mentioned that in FIP2 it was better if your crystal were sort of large crystals. Massive 3 were better for small crystals. So the question was, know your crystal and then ask for ICOS on a certain beamline, or you get the beamline what you get, and then you try to do what you can do.
I think you can do pre-characterization spectra on ICOS offline to see what's possible. We can tune the focal spot of the incoming light and see how it goes.
Well, I have another question. I have many questions to Anton Popov about the microfluidics. All the 3D printings that you're doing, it's absolutely stunning, especially the fact that then you've done one size that can go on your goniometer, so one size fits all. Can we do a rolling application and to try to see which kind of the microfluidics you have is better for a given experiment? So, or do you recommend to try first the microfluidics which are commercially available, and then try to upgrade or optimize for ESRF?
It actually depends, because if you have kind of let's say special task what you want just to try, it's not a big problem for us to help you with what we have actually here. So I think, like, in this kind of user-friendly environment, but if you have some kind of let's say idea for the basis work, you know, like that you want to carry out some particular stuff at the same time with the same results, I think maybe it depends on the task. But of course you can use something which is already on the market. But I would still suggest, if you want to work as I understand, with the more on beamline experiments, or the something in lab experiments?
No, in the beamline experiments.
No, in the beamline I would suggest that we can just have a talk, and then I will try to do as much as possible.
Thank you very much. Thank you.
Thanks to everybody for coming. There's a lot more people than we normally have, probably because it's remote and much easier to do than to get to Grenoble. And I hope you've enjoyed the presentations. We've done a lot of work on the beamlines since we came back after the EBS restart. Not everything's perfect, but we're getting there, I think, and getting there quite quickly. So yeah, so I hope you've enjoyed today's presentations, and I hope to see at least some of you at the ESRF soon. I mean, I realize that a lot of you now will be doing remote access experiments almost exclusively, but it's always nice to see and talk to actual people from time to time. So it would be nice to see some of you at the ESRF and when it's convenient for you. So great. Thanks a lot, everybody. Thank you.